Today we're going to be taking a look at Unit 2, Lecture 6, Gene Expression. And this is a follow-up to the previous lesson on DNA structure and function, where here we take a look at how the information coded in the DNA actually is expressed, which means to manifest in a physical form within the cell. So, for some terminology here, the definition or the way to describe gene expression is the process by which a gene, the sequence of base pairs, is made into a gene product. So what that gene product is some kind of protein and or RNA molecule. Now, from the previous lesson, we talked about genotype and phenotype. Genotype being the DNA information, phenotype being the physical expression or the way that that DNA looks or presents itself. So you can say it in this way, genotype becomes the phenotype. More of a figurative way of saying it, but that's a way to think about it at least. Okay, so generally speaking, what's happening here is DNA is going to be transcribed and then translated, and those two words are going to be described more on this next page here. So we've talked about the central dogma of biology in the past. So if you recall, the central dogma of biology tells us that DNA is ultimately used to make RNA, and then the RNA is used to build protein. Proteins are assembled by individual amino acids being joined together by peptide bonds. Now, in reference to gene expression, transcription is the process by which DNA is converted into RNA. Now, in this particular context, we're actually talking about a specific subtype of RNA called mRNA, which stands for messenger RNA. So the context here revolves around messenger RNA. Now, when we have messenger RNA transcribed, that will then be translated through translation into the protein. And that part involves ribosomes. If you recall from previous lessons that ribosomes, as we described, synthesize protein or build protein. How do they do that? Via the process of translation. So that's a quick sort of recap of the central dogma and then putting that into the context of what we're talking about today. So we're going to start with transcription next. And transcription is, as it says here, the process by which DNA is converted into RNA. Now, what you want to recognize then is that that involves really one primary enzyme. The main participant, as it's often described, is RNA polymerase. And that sounds similar to DNA polymerase. And just keep in mind that these are two separate enzymes, but the name implies that they're similar. And so DNA polymerase takes nucleotides from the DNA template and builds a new structure on top of that during replication. RNA takes the DNA template and builds an RNA transcript, mRNA transcript, on top of that using the DNA as a template. So they're very similar enzymes. The one here referred to as the RNA polymerase. And here you can see that functioning in this figure here. Now, I've given you videos this week. If you haven't watched those videos, I would recommend that you go watch those videos first, and that way you have some visuals, and that way hopefully the written words with the pictures will make more sense to you. So if you haven't watched those videos, I would actually go ahead and stop it now. Go watch at least the first couple videos about transcription and translation, and then I would come back and finish the rest of the lecture. The stages of transcription are just summarized in three general parts: initiation, elongation, and termination. So initiation and termination are what they sound like, the beginning and the end. Elongation is the process by which the mRNA is built using the DNA template. So you can see that here in the middle. The DNA strand will actually open up, and then as the RNA polymerase reads the base pairs, it will then build RNA using the information as its guide. And when it finishes, then it will terminate. The mRNA will then separate, and the DNA will close back together into its original configuration. Now, part of the initiation of transcription is a little more complex than it might sound there. It doesn't actually just begin haphazardly. For the most part, DNA has to be initiated before transcription will begin. And this is a little more complex than what we're going to get into with this class, but we'll kind of touch on the basis here so you at least have an idea of how it works. In the DNA, there are a multitude of different transcription factors, and ultimately these determine what parts of the DNA should be transcribed, and in more complex ways, when and to what extent they should be transcribed. So imagine you have a book of information, imagine a book of recipes. You're not necessarily just going to make every recipe every day of the week. You're going to choose different recipes at different times. And so what transcription factors are controlling factors that ultimately tell you what recipe you want to cook up and for what occasion at what time. And that's the idea behind it there. The way that they work more physically is they will bind to a site in the DNA, and that then allows RNA polymerase to bind on top of that. So the way the RNA polymerase gets its initiation to start is by finding a transcription factor which is bound to the DNA when it's time for that gene to be turned on. Ultimately, without that factor, initiation cannot occur, and therefore these control when transcription begins and ultimately when it is not in play. So ultimately, not only do they allow the initiation, but if you can't begin something, it never begins in the first place, right? Kind of obvious when you say it out loud, but just to state it more clearly in that last bullet point there, these ultimately control the process itself. This is one of the more interesting and more complex ways that scientists are now unraveling some of the mysteries of the DNA. It turns out these transcription factors occur in a multitude of ways, most of which aren't really fully understood. But our DNA is constantly being turned on and therefore by default kept off at times. And as we understand more about the DNA, we start to understand kind of what mechanisms specifically allow these transcription factors to be activated. Ultimately, this figure here, if you want to pause it and look at it, shows you where the binding sites are for these activators, and then ultimately kind of what happens once they're bound, so that we either get transcription, small amounts, or no transcription, depending on what kind of activators or transcription factors are bound to the DNA. And as I mentioned, this is a lot more detailed than what we get into in this class, but that's sort of the idea, at least to give you a general sense of how transcription works in the broader context of your DNA. You have to activate genes through transcription factors. Now, a little bit more specifically, there are sites on the DNA known as promoter regions, and that is really where the RNA polymerase will bind, and it's usually in a close region to where the transcription factors will bind. It's not always in the exact same location, making it even more complicated, but ultimately the promoter is where the RNA polymerase will bind, and ultimately where the transcription information occurs. As I said, often occurs upstream from the gene sequence, not always in the exact location, but one of the things that scientists look for when they're determining where transcription is often initiated are what are called TATA boxes, also known as TATA boxes, TATA boxes. And these are regions in the DNA that are repeating thymine, adenine sequences. And what's interesting about thymine-adenine bonds, remember the complementary base pairing rules, A and T always bind and C and G always bind. The T and A sequences actually have a weaker hydrogen bond compared to the G and the C. So having repeating sequences of A's and T's actually make it easier for the RNA polymerase to separate those bonds because they're actually weaker by comparison to the C's and the G's. So it makes for a physical way for the DNA to separate by having repeating segments called the TATA box. Now, once initiation has begun in coordination with the transcription factors, we now get into elongation. Elongation is the process by which the RNA begins to build, the RNA polymerase, begins to build messenger RNA using the DNA as a template. So it's going to move along the DNA and quote unquote read the information and pair up base pairs in reference to that information. Now, what's interesting is that you have two strands of DNA. Theoretically, you could copy either side. However, if you really pay attention to the way that the base pairs line up and the outcome in the RNA, you'll actually get two different RNA strands with different information depending on whether you're reading one side of the DNA molecule or the other. And so what the cell does is it picks what's called the template strand, which is the right side of the DNA molecule, and that is the side that carries the proper information so that the RNA information is coded correctly. This is one of the mysteries of the DNA, something that's not fully understood. How does the cell determine what side, and how do we know what side? And that's one of the interesting things that we're still learning all the time. What we know is that there is a right side. That's called the template strand. The wrong side, or at least presumably, is called the non-template strand. So these are questions that don't always have clear answers, but what we know is that the cell ultimately will determine the proper side, which is the template strand. And therefore, the RNA code itself is of the right information. Because if you pick the wrong strand, as I mentioned, you'll get a different code. Maybe that code could also work, but it wouldn't be the same if you paired up the template strand compared to the non-template strand. You would actually get two different RNA sequences depending on which side you chose. So the one that is the proper strand is called the template strand. So here you can see a figure showing that the RNA polymerase binds, opens up the DNA, builds RNA using the DNA as a template, and ultimately moves along the template strand, adding the nucleotides in complementary fashion. So if we have a T, we're going to get an RNA adenine. If we have a DNA thymine in the template strand, pairs up with an RNA adenine in the RNA strand. DNA adenine pairs up with RNA uracil. DNA cytosine, RNA guanine. DNA thymine, RNA adenine, etc. Now, if you were to pair, so that's why I was showing you have to have the proper strand, and so if this is the template strand, we'll get AUG, AUC. Now, if you were to pair this on the non-template strand, what you would actually get is that you would get a U here, and then an A, and then a C, and then another U, and then an A, and then a G, etc. So you would end up having UAC, UAG, which would be different than the sequence here. So that's what I mean by the template strand has to be properly chosen, somewhat mysteriously, to make sure that the actual RNA carries the correct information. And that's why we have to differentiate between the template and the non-template strand. Now, termination is being simplified here. Ultimately, there is a terminating sequence where the RNA information ends. We don't want to code too little. We don't want to code too much information. So we have to stop it at the right place. And that's referred to as the termination sequence. This is a little more complicated than what we're showing here. But ultimately, once we hit that sequence, the RNA will then dissociate from the DNA. It will now be separated. And at that point, in eukaryotic cells, it's a little different than prokaryotic cells. But once we terminate the sequence, we now have the RNA separated from the DNA itself. And in eukaryotes, what will happen is that we're going to add a long string of adenines called a poly-A tail. So poly mini A adenine tail at the end of the RNA sequence. And ultimately, what this does is it's essentially a flag, if you will, a signaling structure that tells the cell that this mRNA is now ready to be processed to leave the nucleus. Without the poly-A tail, that RNA sequence that we just transcribed will actually be maintained in the nucleus, and it will not leave. So the poly-A tail is an additional level of complexity that tells the cell this RNA piece that we just created is now ready to leave the nucleus. And presumably what this does is it allows the cell to transcribe RNA and then maintain it in the nucleus if the poly-A tail is not added. So that way certain messages can be transcribed and then held, or transcribed and then released via the addition of a poly-A tail. So there's a little more variability there that the cell has control over. Now, once the RNA has left the nucleus, it will then be in the form of messenger RNA, mRNA, as I mentioned earlier. And what the term messenger implies is that we actually have a message in the RNA. You might think of it as a code. And ultimately, the individual parts of that message are actually made up of what are called codons. And that's where the term code can be used a little bit more directly. So the message is made up of codes. And so therefore the term codon. What a codon is it's a sequence of 3 nucleotides. And as I'll show you in the next figure here, the mRNA essentially consists of a long series of codons. Each codon consists of 3 nucleotides. And in reference to the code itself, what we actually have is the 3 nucleotides coding for amino acids. So think back to the whole process. We're trying to build protein. What are proteins made up of? Amino acids. So how do we determine in our protein what amino acids we want? We determine what codons we have in our messenger RNA. So as we build the code, we're actually building sequences of codons, each codon being an individual sequence of 3 nucleotides. Now, at the end of that sequence then is a special variation of codon called a stop codon. And rather than coding for an amino acid, this is one of 3 unique codons that instead codes for a stop message. And the next figure here, the next slide, has a figure that will represent that better, more visually here for you. So this is the figure here. So when it's said that DNA codes for protein, it's more specific to say that DNA codes for the amino acid sequence of a protein. And that's what we're referring to here in terms of codes. So if the mRNA is sequenced with this particular lettering, what we can actually see when we break it down is that the letters are grouped in units of 3 called codons. Codon 1, codon 2, codon 3. Now these codons then each as a unit of 3 will go on later to signify what amino acid we need and in what sequence, which one comes first, which one comes second, third, etc. And ultimately, how many. Some proteins are relatively small. There's proteins called peptides that are essentially small, short sequences of proteins, maybe only a dozen or 2 dozen. And then you have small proteins that may be 50 to 100. And then you have maybe average proteins closer to 300, large proteins maybe closer to 1,000 or more in reference to the number of amino acids. So not only do we need to know what amino acids to use, what sequence to put them in, but also how many we need to have. Is it a small or large or intermediate sized protein? So that's what is all carried in the messenger RNA, these literal codes that code for the amino acids that ultimately tell us the type, number, and the order of the amino acids. This, by the way, tells us that in our DNA, we have a literal code, basically a language, in which the cell is communicating from one part of the cell to another. Here's what I have in my information. Here's how you're going to take that information and assemble it into a physical structure, which is the protein. And as we've talked about previously in metabolism, the overall shape of the protein determines its function. And that shape of the protein is a direct consequence of the amino acid sequence, which is a direct consequence of the codons, which is a direct consequence of the DNA nucleotide sequence itself. So it's fascinating. In your DNA, you actually have a literal language that tells your cells how to assemble proteins in reference to sequences of amino acids. Amazing complexity, by the way. This really has only been elucidated going back to roughly the 1970s. The DNA structure of the DNA structure itself was elucidated in the early 1950s, and then the functionality became more obvious between the 1960s and 1970s. So this is something that's, maybe only 50, 60 years old in terms of our understanding. Very, very interesting stuff. Now, codons themselves consist of 3 individual nucleotides, A's, U's, C's, and G's, if we're talking about the RNA language. So remember that uracil replaces thymine. So if you have 4 letters, A, U, G, and C, and you can arrange them in 3 different positions, 3 U's, CUU, CUC, all these different combinations. If you do the math, what you can say is that you have 4 letters that can be taken to the 3rd exponent. And if you do 4 to the 3rd power, you'll get 64. And it turns out there are exactly 64 codons, which is the maximum number possible with 4 letters arranged in 3 positions. Of those, we have one special start codon, which always goes at the very beginning of the sequence. And then we have several designated stop codons, which always occur at the end of the sequence. Everything else will occur variably in between from any of the other codons that you see here, and they each code for one of the 20 different amino acids. And what you're looking at here is effectively the code of life. This is effectively the way that life's code can be visually represented. On the left here, you see the various combinations that we can put together in 3 base pair combinations. So we could put just 3 U's, or 2 U's and a C, or 2 U's and an A, etc. And depending on what exact sequence we put together, we will end up coding for one of 3 things, either a start sequence, a stop sequence, or any of the amino acids in between. And what this code here tells us is what we expect to get whenever we have a specific combination of letters. And what's fascinating then is that during the next part, translation, the ribosomes miraculously read this information and use that as a building code to assemble amino acids. And that's what you're essentially looking at here, is how the cell determines what to put together and how to put it together based upon a literal genetic code. I'm not going to make you memorize this. Just be familiar with what you're looking at. You're looking at codon sequences and what they code for. And these are the amino acid names if you're not familiar. Phenylalanine is one of the 20 amino acids. Leucine, serine, proline, threonine. Now, another thing that's worth pointing out here is that there's what's called redundancy in the code. So redundancy means that we end up with several different codon sequences that often code for the same amino acid. So UUU and UUC both code for phenylalanine. And what that allows for is a little bit of variation, very often in the last letter. So if I have a mutation, for example, and it changes this letter from a U to a C just by chance, I can still end up with the same amino acid. So there's what's called redundancy and then wobble. Redundancy is the idea that multiple codes code for the same amino acid. And wobble is the idea that I can sort of get a close matching code and still get the same amino acid. So if I have UUU or UUC, I'll still get phenylalanine. So some of these have multiple variations. Leucine has a total of 6. Phenylalanine only has 2. Serine has 4. Proline has 4. Threonine has 4, etc. And so that's where you can kind of start to look a little more closely. Histidine, glutamine have only 2. There's an interesting, seemingly random variation here, but there's an actual mechanism here by which the cell can mitigate mutational effects and also increase efficiency in the translation phase. That part's a little more complex, but that's why you see multiple codes all coding for the same thing. That's referred to as redundancy, which gives rise to what's referred to as wobble. And those are mentioned in the textbook, although I don't have those terms here. Now, once the mRNA sequence is complete, it has its poly A tail added to it, it will then be allowed to exit the nucleus through the nuclear pores. Those are key structures in the nucleus itself. And then from there, it'll be shuttled into the cytoplasm, really more specifically the rough endoplasmic reticulum in a eukaryotic cell. And this is where we'll find the ribosomes. And the ribosomes then allow us to begin the next process, which is translation. So following transcription, the next process is translation. And this is the conversion of the mRNA into the protein. But more specifically, what we're doing with translation is we're building the chain of amino acids, which can be referred to as the primary protein sequence, can also be referred to as a polypeptide or a polypeptide chain. So ultimately what we're doing here with translation is attaching amino acids together to form a peptide or polypeptide chain, also known as the primary protein sequence. And we're doing that not randomly, but based upon the information in the mRNA, which is based upon the information in the DNA. So we have a code, and the ribosome is now going to assemble that code into a physical manifestation, which is a protein. And I will tell you, in my opinion, of all the cellular processes in all of biology, this is the most mysterious and the most difficult to really wrap your head around. It seems sort of odd and it doesn't seem possible, but it is happening, albeit mysteriously. And really, what I'm showing you right now is science best attempt to explain what's happening here. And it definitely happens. It's just whether it works exactly as we're seeing it here. But the outcome is predictable, and so we're looking at the best estimation of how this process works. So we're building a protein sequence. This occurs with the ribosome. Just to clarify, this red structure here represents a ribosome. Ribosomes occur in two parts. You have the large subunit here and the small subunit here. And what we're showing is a segment that's cut in half. In other words, the ribosome is represented being cut open so you can see down the middle of it. And so what ends up happening is that the RNA will sandwich through, as you see here, and then another form of RNA, as we'll talk about on the next slide, called transfer RNA, which looks like this here, brings in the amino acids shown here, and then allows those to be assembled together into the chain, as you see here. This occurs in both prokaryotes and eukaryotes, depending on what type, a eukaryote or a prokaryote, will determine whether this occurs either in the rough ER, in eukaryotes, or in the cytoplasm, strictly in prokaryotes. But in eukaryotes, it can actually be done in the rough ER and in the cytoplasm, in both locations. But either way, in prokaryotes and eukaryotes, the same process takes place in a very, very, very similar fashion. So for visualization here, we have our DNA segmented in genes. Each gene then can be represented by a series of nucleotides. Taking the template strand, we then build an RNA and mRNA sequence. Each mRNA sequence can be divided into codons, and those codons ultimately break down into amino acids. And so as the ribosome reads the RNA, the mRNA, it assembles the amino acids based upon the coding information. This allows us then, as we put amino acids together, to assemble the primary protein sequence, also referred to as the polypeptide sequence. Now, in protein chemistry, what will then happen is that chain will then bend and twist and fold, and sometimes multiple chains will be joined together, and ultimately that final product will be determining the protein function. So usually the function is not fully manifested at the time of the end of translation, but rather the initial portion of the protein that folds and then later gets its functionality. The major participants in translation are the product of transcription, first and foremost, the messenger RNA. So that's essentially the product of transcription, and now one of the beginning points for translation. The next are called transfer RNAs, tRNAs. And the name implies what they do, so they transfer. What do they transfer? They transfer amino acids. So this is essentially the workers of the translation process. I always imagine it like little workers bringing bricks individually to build a brick wall. They effectively bring one amino acid at a time and will have as many of those as required to build the entire protein sequence. So each amino acid requires a separate transfer RNA, and thus many transfer RNAs are required for just one protein. The other major participant are the ribosomes themselves. Ultimately, ribosomes facilitate this process and allow the combination of the mRNA and the transfer RNA to come together within the ribosome. So I think of the mRNA as the code, transfer RNA as the workers, and the ribosome as the facility where the information and the workers are brought together to build the final product, which is the product of the facility, which is the protein itself. Transfer RNA, since we haven't talked about that yet, is the next thing here. Transfer RNA is an interesting molecule. It starts out kind of like mRNA as a long chain, but then it folds into this weird, what's often referred to as a hairpin loop structure, or sometimes called a clover structure. These are just arbitrary terms. But see this linear, you can almost see how this is a linear structure rather. If you took this entire linear structure and broke all these little bonds shown here, these hydrogen bonds or these little lines, if you snapped all those hydrogen bonds, this would unfold into a long linear sequence, kind of like mRNA. But with transfer RNA, the codons are, we actually don't have codons. We have a different process here. And the way that the RNAs are sequenced allows for hydrogen bonding to occur such that the linear RNA folds into a more three-dimensional, or shown here in 2 dimensions, a structure that looks kind of like this. So this is a 2-dimensional model, and this here represents a slightly more accurate 3-dimensional model. So within this structure, then, what we have are 2 key regions. One is called the amino acid attachment site. That occurs in this proportion of the molecule up here. The other region, more confusingly, is referred to as the anticodon region, and it occurs in the bottom sequence down here. Now, the term anticodon sort of implies what it does, but doesn't really state it or doesn't really make that obvious. But if you recall, mRNA carries the code, the codons that code for amino acids, and we need to build a protein that is specific to this code. We can't just have random amino acids showing up at any place. So we're not just pulling amino acids at random. We need to pull specific amino acids. So the way the cell organizes this is that it facilitates the combination of anticodons in correlation with the amino acid at the top. So then what happens is, in the ribosome, wherever the mRNA contains a codon, the transfer RNA will need to have a matching anticodon that has a base pair complementary to the codon itself. So anticodons pair up with codons with complementary base pairing rules. And I'll show you an example of that, a couple of them here. I'll let you write that down here, and then I'll show you some visuals to help you understand that a little bit better. So this all happens inside the ribosome. And inside the ribosome, we will have a transfer RNA come in, and the anticodon sequence can be represented down here as an example, CUC. Up here, we have an amino acid carried at the amino acid attachment site. Now, when the transfer RNA and the mRNA come together, what we need is a complementary set of codons with anticodons. So the complementary base pairing rules between RNA are such that a RNA guanine will complement with an RNA cytosine. An RNA adenine complements with an RNA uracil, and vice versa. So if I have a codon that says GAG, the complementary anticodon would be CUC. And if those two match at the site of the ribosome together, that signifies that this transfer RNA matches this codon. So if I now go look at my codon chart, what I can see is that the codon itself, GAG, is complementary to CUC, which is on the anticodon. And amazingly, what will end up happening is that the codon in the RNA, the mRNA, has to not only be complementary with the anticodon of the transfer RNA, but then, going back here, the transfer RNA will actually carry the amino acid, signified not by the anticodon, but by the actual codon itself. So what that means is that if I have an anticodon with CUC, it will actually have the amino acid glutamic acid, which is coded by GAG. So in the code, I have GAG, which tells me I need glutamic acid. So what that means is that when I bring glutamic acid in, the way that we check that code is through the anticodon complementary base pairing. As long as this is complementary, that means that it carries the proper amino acid, which is signified in the mRNA. So the anticodon doesn't actually carry the code, the mRNA codon carries the code. And as a result, we'll carry the proper amino acid up here. So now when we take a look at this figure, which is a representation of what's happening inside the ribosome, we're kind of cutting the ribosome down the middle so that we can see what it would look like inside. We see the mRNA lined with codons, in the transfer RNAs with their anticodons. And as I mentioned before, it's a mysterious process. It's happening inside the cell in ways that we can't fully see and visualize, so we have to kind of take it with a certain level of indirect evidence that this is happening. But what is presumed to be happening, and what we sincerely believe, is that the transfer RNAs come in, they line up, and as long as they match, that means that the code is properly orientated at that point. The amino acid, therefore, is correct, presumably, and it will then form a peptide bond with the elongating chain that has formed from the previous transfer RNAs. So effectively, you'll have two spots for the tRNAs to come in, and then an exit spot. So as they come in pairs, one amino acid transfers to the next. That begins to grow a chain. Then the amino acid that previously donated is kicked out. A new transfer RNA comes in, and that process repeats. And this can happen at a pace of several dozen amino acids per second. So it's a relatively fast pace. And as I mentioned earlier, there's redundancy and wobble. So what that means is that we can have a little bit of error, especially in that third base pair, where, for example, looking at glutamic acid, both GAG and GAA will both fit even if they're not precise. So even if that A or the G, for example, is variable right in that spot, then we can still get the right amino acid, glutamic acid. And so that's what's referred to as the wobble, which is the idea that you have a little bit of room for mistakes or variation in that third base pair. So now that you understand the way that the code is translated, we can see that there is an initiation, and this is almost always occurring with the start codon. If you go back and look at that chart, there is one codon for formal methionine, which is AUG. And this is pretty much always the one that begins the process. So initiation kind of forms around that particular transfer RNA, and this is one of the reasons why they call it formal methionine. Ultimately, the ribosomes sort of sandwich around the mRNA. We're leaving out a little bit of detail there to keep some of the simplicity here, but ultimately we have a formal initiation process around a particular start codon. Elongation is the process then by which each amino acid is brought into the growing chain, as we just showed, matched with the codons and anticodons, and then a peptide bond forms, and that will occur for however long the amino acid chain or the polypeptide is supposed to be. Termination then occurs with one of three formal stop codons. Looking at that chart again, you can see that those are highlighted in red. And so you have either UAA, UAG, or UGA as your official stop codons, which will always occur at the end of the official codon sequence. At that point, rather than having a transfer RNA, this will attract release factor, an additional protein, which is now required to disengage the mRNA from the ribosome, split the two halves of the ribosome back apart, and everything is released. And at this point, the actual polypeptide is formed. So, as we said before, the product is a primary protein sequence, which can also be referred to as a polypeptide chain. And it looks like this here to a certain extent. At that point, you have an unfolded three-dimensional state, or the polypeptide is in an unfolded three-dimensional state. It's not quite ready to function at that point, and this goes outside the scope of this particular lecture, but ultimately further folding is required before it will achieve its full three-dimensional state, as I mentioned earlier. So to kind of summarize that in one figure here, the entire process of gene expression, DNA opened, RNA polymerase transcribes, mRNA is now translated into a polypeptide. Now, one of the things that's happened in recent years is scientists, having understood this process, have realized that you can actually kind of take you can get an idea of what's happening in a cell by figuring out what mRNA sequences are being transcribed at any given moment. Understand that cells are highly variable. Even cells with the exact same genetic sequence, such as all the cells in your body, are going to be utilizing the genetic code in a variable way. So right now your muscle cells presumably are using the genetic code differently from your nerve cells, which are different from your skin cells and your hormonal cells, etc. So the transcriptome then is a way for us to kind of take a snapshot of what are the RNA molecules being produced in that cell at that specific time under those specific conditions. And by analyzing that, you can start to gauge information and insight as to what kind of information processing is happening. And you might actually be able to determine that under this physiological state, the cell is producing these mRNA molecules, but under a different state, it will produce different molecules. This could occur in a variety of ways. So it basically gives us an idea of what genes are being expressed. So we can in some ways see what the genes are by looking at the DNA. What is the genome? What are all the different individual sequences? And then we can look to see how are they being expressed, which is going to change presumably even from moment to moment, or I should say potentially from moment to moment. This is one of the areas of active research. This is an ongoing investigation, much more questions than answers. But one of the things they found is that, as I mentioned, different cell types and tissues are going to have very different gene expression. And that's actually one of what makes them different. They all have the same genetic sequences, but they use them differently. This also occurs at different developmental stages. When you're growing inside the mother's womb, you're going to be turning on different genes compared to when you're a teenager. And that's going to be different compared to when you're an adult versus later in life. So different developmental stages will have different activities in reference to gene expression. And then environmental conditions. One of the things that they've looked at is how stress affects the transcriptome. How does different diets affect the transcriptome? And I don't think there's a lot of clear answers there, but they're investigating those things, trying to get more and more insight. So ultimately, it reflects a dynamic nature of the cell, the idea that we have a code, but it's expressed variably. Ultimately, this is an area for further research, as I said, and over time, the hope there is that we understand more and more specifically how the cells respond in these different environmental conditions or different stages. And by the way, this could also relate to how we get something like cancer, for example. This figure here is just a reference to all the different cell types, or at least many of them in the body. And so, for example, stem cells, bone cells, blood cells, even cancer cells, as I just mentioned, all those cells in your body have the same DNA, but they're all undergoing a different process of transcription. So taking a snapshot through the transcriptome helps us understand why these cells are different, and especially in relation to cancer, what exactly is going on and why are these cancer cells dividing and growing and spreading in an uncontrollable fashion. There's a lot of hope and a lot of potential here to treat cancer, and in fact a lot of that is already in the early stages of development and even research. Last few slides here I want to go through are alterations of gene expression. So gene expression, as I just mentioned, is variable. One of the things that can change it all the way back to the code itself is a mutation. And this is more of a fundamental understanding that we've had about genetics for quite a while. Mutation is essentially a change to the code itself. And when you change the code, you at least have the potential to alter the final product. In that same sense, then, you have the potential to change the shape and the function of the proteins that are the product of translation. I'll have you read more about this in the book, different types of mutations in reference to point mutations. Not every mutation leads to a change, such as a silent mutation, for example. But mutations often have a high degree of potential to make a change. But that actually occurs all the way back at the DNA level itself, such that the mRNA is altered and then the translation of the protein is altered. And that's been our fundamental way of thinking about gene changes for many decades. A more modern, newer aspect of that, however, is epigenetics. And epigenetics is actually the change in the pattern of gene expression and not necessarily the change in the information itself. So the information can stay the same, but epigenetics is a change in how we express it. So, for example, the timing. Genes can be expressed slowly, they can be expressed very rapidly. Genes could be expressed in small amounts. You might make only a small amount of protein, or you might make lots and lots of protein. And you may express genes only in your muscle cells that never get expressed in your nerve cells, vice versa. So these are aspects of epigenetics where we try to understand what is it that's making those differences occur. Epigenetics then is described as changes in gene expression, and without alterations in the underlying DNA sequence, to kind of put a slightly more specific definition to that here. You can inherit some of these changes. However, they can also be reversible. So there are aspects of your lifestyle, for example, environmental conditions, and different stages of development, as I mentioned previously, whether you're developing in the womb or in your early toddler or teenage years, those factors will all be different. Now, the way epigenetics works is at above the genome level. So epigenetics is a word that kind of translates loosely to above the genome. And so what's happening here is that it's actually a change in the organization of the DNA, which has to do with the way the DNA is packaged up and bundled together. And this is also shown in one of the videos I have you watch. One of the last videos is a great visualization to kind of give you an idea of how DNA is condensed and bundled and organized. So the visualization in that video there is to kind of help you understand this particular segment a little bit better. But as I'll show you here with the picture, a couple of pictures, the DNA is essentially wrapped and organized, and that wrapping prevents the DNA from being expressed, and therefore to express it you have to unwrap it. So you can wrap it up tightly to prevent expression or unwrap it to allow expression. And although that's an oversimplification, that's basically what epigenetics involves. So DNA starts out as a form called chromatin. Chromatin is essentially a long linear form that's wrapped around histones, which are then further bundled into nucleosomes. And at this level, the DNA is packaged in a way that makes it difficult to actually get down into the individual base pairs and to actually access the genetic information. And the way I kind of think about it, it's like having a book of information, but the book is closed, and therefore you can't read it. So to read it, you have to unwrap the DNA, just like you would kind of open the pages of a book. So this is where it gets a little more technical. I'm not going to make you memorize each one of these little terms here, but histones are proteins that the DNA initially wraps around, and then nucleosomes are bundles of those histone proteins that occur in packages of 8. And from there, then we get kind of more and more units that then fold together to make chromatin, which are thread-like structures of DNA that then later will go on to form chromosomes, which people often are much more familiar with. Chromosomes, however, only form right before cell division. So if a cell is getting ready to divide and split to make a copy of itself, that's actually the only time that chromosomes form. So ultimately then, the DNA is packaged up, and as I mentioned, you can't actually read the information when it's all bundled up in the form of chromatin. So what we have to do then is essentially lead the cell to either open or close the DNA. And this is done through one of two ways: DNA methylation or histone modification. So DNA methylation is the addition of methyl groups to the actual DNA molecule itself, as you can see over here. Me stands for methyl, and it's actually a little chemical group that's physically bonded or attached somewhat weakly to the physical DNA strands themselves. Now, these are reversible and they can be added and removed. So once a DNA molecule is methylated, that will affect the way it is either wound or unwound, or therefore transcribed or not transcribed. The other way that it can work is at the histone level, and histone modification then is a similar mechanism to allow for the winding or the unwinding of the DNA. So ultimately, the main idea is that these two mechanisms, methylation and histone modification, will either allow loosening or tightening of the DNA. Loosening allows for transcription, tightening prevents it. And this is an exciting new area of research that is allowing scientists to go beyond the initial understanding of mutation and into a new area where we see that the genetics of the cell is even more complex than previously thought. And we see now that often when cells are not functioning properly, at least often, that can be translated or correlated back to the genetic level, and now often being seen with these epigenetic disorders that are out of whack with the normal functioning of this loosening and tightening process. All right, we'll stop it there. Have a great day.